day, this is Professor Will, CBMD PhD. And today's Friday, December 3rd, 2021. It is 9:54 and 25 seconds, a.m. Eastern Standard Time. And today's research, biological cell science. So let's hop to it here. Harnessing the organization of the cell surface. Date December 2nd, 2021. Source, ETH0. The scientists have developed a new method to determine how proteins are organized on the surface of cells. Insights gained with the technology could lead to the development of novel drugs to fight cancer. Biological cells have multiple functions, and they need to communicate with each other to coordinate them. Molecules on the cell surface are central to this process. For decades, biologists have been studying such surface proteins, and it is becoming increasingly clear that not only their presence, but also their organization on the cell surface is crucial to the function of the cell. The proteins aren't simply distributed evenly and independently of one another across the cell surface. Instead, they are organized into molecular communities. In these communities, proteins often work together to fulfill cellular functions. And with this technology called LUSMX, the researchers can determine with nanometer scale precision how proteins integrate into an organization on the cell surface. In other words, which proteins are in proximity to each other. And so far, scientists were able to measure interactions of individual proteins that have a high affinity for each other, as well as for molecules that reside inside the cell. However, the new method is the first to enable scientists to specifically detect the organization of the entirety of the cell surface molecules. Researchers refer to this entirety as a surfaceome. The term is composed of the word surface and the suffix ome. That is also used in terms such as genome or proteome. Researchers specifically modify a particular surface molecule so that it likes to give kisses to molecules in its proximity, and then they check the other surface molecules for traces of lipstick. And in more technical terms, a small chemical compound is attached to a protein of interest. When irradiated with light, this compound produces small amounts of what are known as reactive oxygen molecules that oxidize surface proteins in the immediate vicinity. And using a specific enrichment method and mass spectrometry, in combination with statistical data analysis, the scientists can ultimately identify which molecules have been oxidized. And to determine the distance between the protein of interest and other molecules, the researchers repeat their experiments under slightly modified conditions that affect the amount and survival time of the reacting oxygen molecules. And these include the length of irradiation with light and the choice of medium in which the cells are cultured. The more reactive oxygen is locally generated, and the longer the process continues, the wider the area in which surface molecules get oxidized. Researchers will now use the technology to compare cells from healthy and diseased people. They are trying to understand how a disease changes the organization of proteins at the cellular level. For example, when a healthy cell transforms into a cancer cell. The scientists are in the process of creating a reference map of healthy cells. They then plan to use it to identify differences in the organization of protein communities on the surface of diseased cells. And knowing which molecules exist on cell surfaces and how they are organized can become significant in areas such as the development of novel drugs to fight cancer. Modern cancer drugs employs a cell-killing agent is often coupled to an antibody that recognizes a surface molecule that is present in large quantities on cancer cells. And this means that cancer cells are killed with a reasonable degree of specificity. However, many cancer-specific surface molecules are also found on healthy cells, albeit at lower concentrations, provoking these drugs to also kill some healthy cells. And if two molecules were found to be adjacent, only on a disease but not on a healthy cell, drugs can be developed that recognize these two molecules together. The drug would then kill a cell only if both molecules were present on the cell and adjacent to each other. And this is precisely the information that the new technology provides. The scientists and researchers show that the scope for using this method goes beyond the investigations of which cell surface molecules are next to each other, and they also tag viruses and drugs with a small chemical compound that produces reactive oxygen, and this allows researchers to study the binding events of viruses or drugs to cells. Furthermore, the method makes it possible to investigate which protein communities are involved in the interaction between two different cells. The scientists demonstrated this using the example of communication between immune cells. And in this new way, the new method can help to understand how drugs work and how viruses or immune cells recognize other cells. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.